So, so this is basically a continuity in from the previous lecture. Okay, previous lecture we discussed about fertilization implantation, and basically this is also a part of embryology and some part of physiology, fetal physiology. Okay, so I think this is just a revision class for you all because you have already gone through the embryology in your basics. So briefly I will talk about fetal development, fetal physiology, and I will conclude with the circulation. Okay, so today's learning objective: overview of the fetal development, which follows the fetal physiology, and I will conclude with fetal circulation. Okay, so basically fetal development, initial part of pre-embryonic phase we discussed. Okay, so from the fertilization, okay, from the ovulation till the fertilization, and from fertilization formation of male and female pronucleus, then zygote, okay, and the division of cleavage stage started. Okay, and around day 3, day 4, the ball of cells, what we call morula morula stage occur, and morula is just collection of the cells, okay, that we call blastomere, but with still the morula phase but still there is zona pellucida is still intact there. Okay, and from morula onwards, yes, blastocyst, when it enters the cavity excuse me. Okay, then morula, it moves to the uterine cavity, and there this zona pellucida, okay, this becomes a bit porous, so the fluid enters and the cell gets segregated. So one pole it is confined as embryonic pole, and periphery it's that is, yes, trophoblastic layer, okay, which give rise to future placenta. Okay, and and that stage is called blastocyst stage. Okay, and blastocyst it get implanted into endometrium. Okay, this should have. Okay, so after that, so then it starts the stage week 2, and week 2 we know week 2 is a rule of 2. Okay, so these collect some cells, they divide into embryoblast and trophoblast. Embryoblast it give rise to future embryo, and trophoblast it gives rise to placenta. Okay, and there is a 2 cavity, so yolk sac formation as well as amniotic cavity. Okay, and in week 3 it follows the embryonic period. Okay, so let's see. So this part from week 1 to week 2, so this part we discussed already. Okay, so from zygote to morula, blastocyst, okay, and subsequently embryonic disc, bilaminar disc, okay, and formation of early uteroplacental circulation by invasion of syncytial trophoblast, and there is formation of primitive streak. From there the cell move, okay, and the process is called gastrulation. Okay, and all the cells they move, and ultimately they give rise to 3 layer of cells. Okay, that is trilaminar disc, and it starts the week 3. Then first organ to form is initially there is formation of notochord. Okay, and it induces the formation of central nervous system. Okay, usually neural tube, and neural tube give rise to neural vesicles. Okay, and so if there is any defect in the closure, it can cause neural tube defect. So let's start with, so embryonic period is, so it start from week 3 to week 8. Okay, so this is the main embryonic period. Okay, now initially and subsequently all the organs develop, okay, from week to from 8. Okay, that's why we call it embryonic period. And after 8 weeks, it is the fetus is recognizable human. Okay, there now just further maturation of the organ occurs. Okay, so all the organs have almost formed by week 8. That's why from 9 week we call it fetal period. Okay, this is the post conception. Okay, if you do the menstrual dating, then you have to add 2 weeks, right? So from 5 to 10. Okay, from 11 to 40 weeks we call it fetal period. Okay, so remember, so this is the post conceptional gestation age. And we also briefly discussed about some teratogens last time. So these are basically chemicals, okay, which chemicals or virus, okay, any drugs which can affect the fetal development, okay, which can cause congenital anomalies. So which part is the most sensitive? As we all know, there is a rapid development. Okay, the cells are moving and they are making all the organs. Okay, so that's why this is the phase of rapid development from week 3 to week 8. This is embryonic period, and this is very susceptible for teratogens. Okay, so you can see here, so this the purple one. Okay, so this is the highly sensitive period, and this is less sensitive period. Okay, for neural development, you can see up to 16 weeks. This is the highly sensitive period. Okay, so the teratogen can affect up to 16 weeks. So initially, okay, so within almost 3 to 5 weeks, this time frame, okay, if there is defect in the closure of neural tube, it can cause neural tube defect. Okay, so as early as 3-4 weeks there could be neural tube defect, and later on, because the insult from the teratogen, it can lead to mental retardation. Okay, and so then second is a heart. Okay, in the formation of heart, you can see, so up to week 6, this is also very vulnerable period. So if teratogen can affect, it can lead to bronchosarcoidosis, atrial septal defect, and ventricular septal defect. Okay, and after 6 weeks, this is less sensitive, but still can affect. For upper limb, up to 5 weeks is sensitive period, so it can cause amelia for meromelia. Okay, so this is defect in the limb, seal-like limbs. Okay, so mainly the teratogen which causes this is thalidomide. Okay, okay, so this is basically, so these two amelia, meromelia, okay, this is caused by thalidomide. Another is okay, regarding the face, cleft lip, up to 6 weeks it is very sensitive. Okay, and ear up to 9, almost 9 weeks, rosette, okay, malform ear. Okay, similarly, eyes up to 8 weeks, teeth staining up to 8 weeks, and cleft palate. Okay, so palate is bit late than lip. Okay, so lip is a bit early, and so palate up to 9 weeks. Okay, and look, regarding the teeth and eye, up to 38 weeks, it can affect any time. Okay, so that's why we do not give tetracyclines. Okay, because they got staining of the teeth. Okay. Similarly, masculinization of female genitalia. If the mother is taking some androgens or androgenic drugs, it can masculinize female genitalia. Okay, so mainly it affect around 8 up to 9 weeks, but it can affect any time. Okay, so so what we see, the major defect, okay, major congenital anomaly, usually it occurs during the embryonic period. Okay, but a minor defect can still occur throughout the gestation. Okay, so basically this is the take home message. So let's discuss briefly about the embryonic development. Okay, post conception from day 14 to day 21, notochord develop. Okay, so after the trilaminar disc formation, there forms the notochord, and it induces the what it induces? Yes, neural tube development. Sorry, yes, neural tube as well as formation of central nervous system. Okay, so notochord develops and ectoderm thickens to form neural plate and neural fold. Okay, From day 21 to day 28, so week 3 and to week 4, the neural fold they fuse to form the neural tube. Okay, so primitive, and it also primitive heart. Okay, primitive cardiac chambers. So first heartbeats around 3 weeks. Okay, so that is post conceptual around 5. Okay, so it is basically 3 to 4 weeks, 21 to 28, and in post menstrual dating it becomes, yes, week 5 to week 6. Okay, so basically a neural tube closure, it occurs, I think it's day 20, day 24, and anterneural pore, and day 27 is posterneural pore. Okay, so if there is any defect, anterior neural pore defect, it can cause anencephaly. Posterior defect can cause spina bifida. Okay, from week 4 to 6 post conception, optical vesicle occur. Okay, so complete neural tube closure it occurs by 30. Okay, so it's written in 30 year. Okay, so it ranges from 27 to 30. So it is completed by 30. Okay, day 30, and limb birds appear and formation of face. So as early as 6 weeks post conception, there is formation of face. Okay, and limb bird appear. From week 6 to 8, all the major structure form. Okay, so complete ventricular septum and recognizable human. Okay, recognizable human by almost 6 to 8 weeks post conception. Okay, and from 8 to 12 weeks post conception, okay, when the embryo size is around 30 to 60 millimeter, this is external genitalia develop. And this embryo size we call it crown rump length, because we measure from the crown to coccyx. Okay, so this is crown rump length. Okay, and this is the, in ultrasound we measure CRL. Okay, so this is the crown rump length, and basically this is the size of the embryo. Okay, so 30 to 60 millimeter of embryo, the external genitalia develop. Okay, and from week 20, the skin. Okay, skin is covered by lanugo. Okay, and vernix caseosa is also present. And week 28, testes descend into the internal inguinal ring. So baby is viable. So independent survival after delivery, after 28 weeks. Okay, so that is the viability. And week 36, one testicle descend into the scrotum. Okay, and lanugo tends to disappear. Okay, and week 40, both testicles descend into the scrotum, and the nail they project beyond the fingertips. Okay, and posterior frontal are closed. Okay, so this is all about the summary of the embryonic development. So last time also we discussed a bit about trilaminar disc and ectoderm, mesoderm, and endoderm. Okay, so ectoderm, they mainly form the external structures. Okay, like skin, integumentary system, as well as neural nervous system. Okay, in middle layer, this is the sandwich layer. Okay, so middle layer it forms the muscles, bone, blood vessels, and the inner layer it forms the all the lining, GI tract lining. Okay, so these things, so we discuss. And so basically from 2 week, okay, week 2 to week 3, then series of event occurs. Okay, there is differentiation of cells and formation of the organ occurs. Okay, and also the main things to bear in mind in this slide is, remember the embryonic period. Okay, so embryonic period, so this is very vulnerable for teratogens. Okay, so this period is very vulnerable for teratogens mainly, though it can affect fetal period as well. But this initially pre-embryonic period, okay, it yes, it's about all or none rule. Okay, what is all or none rule? If the teratogen affect, it can damage the germ cells and there won't be any further development of the fetus. But if there is no damage occurs, then it will survive. There won't be any effect because the embryo hasn't started differentiating. Okay, so that's why in this first pre-embryonic stage, okay, or post-conception stage up to week 2, all or none rule occurs. Okay, regarding the teratogens. But from week 3 to 8, this is the major teratogens. Okay, so major congenital anomaly occurs by 8 weeks. Okay, and from 9 to 38, minor defect occur. Okay, as well as functional, like CNS patient may develop mental retardation, slow mentation. Okay, so like that. So this is all about the fetal development. Okay, so now let's discuss about the fetal physiology. Okay, so in fetal physiology we'll discuss about nutrition, how the fetus get nutrition. Okay, so first in the embryonic part, okay, within the tube, so there is adsorption. Okay, initially during the fertilization and immediately in the cleavage stage, all, okay, yeah, these blastomere, they get nutrition from cytoplasm. Okay, then it absorbs from when it moves, morula moves to the uterus. Okay, in blastocyst it absorbs from the uterus. Okay, and till it gets the uterine placenta circulation, it is from the uterine and tubal secretion. Okay, and then finally by the, once the uterine placenta circulation is established, and finally it gets from the blood all the oxygen, nutrients from the mother, it exchanged, okay, by the chorionic villi. We know as early as day 13 there is formation of primary villi, then around 16 formation of secondary, and by almost week 3 there is formation of tertiary villi. So formation of tertiary villi, there is exchange. Okay, so we'll discuss a bit about that. Then fetal blood. Okay, so when does the fetal blood appear, we'll discuss that in urinary system, skin, GI tract, respiratory, and fetal endocrinology. Okay. So remember, so first is absorption. Okay, so post fertilization up to week 2 there is absorption from tubular uterine secretion. Then histiotropic transfer. Histiotropic transfer means, okay, this is basically from the cytoplasm. Okay, it's not from the mother, it's from the cytoplasm. Now this is from the mother, but the secretion is from just diffusion. Okay, decidua. Okay, mainly by diffusion, and later on by the trophoblastic lacunae blood. Okay, so mainly this trophoblastic lacunae blood, this is basically a hematotrophic transfer. Okay, so mainly an histiotropic means the tissue transfer. Okay, so before uteroplacental circulation, it is by the diffusion. Okay, so it's mainly by diffusion. And finally is the hematotrophic. Hematotrophic, as the name suggests, it's from the blood. Okay, the nutrition is get from the blood. So once the active and passive transfer of the fetal circulation is started, once the fetal circulation is started. Okay, so it's by week 3. Okay, week 3 and 4, they would the hematotrophic transfer started. Okay, and this is the main form, and it continues, and placenta is the main organ which supply all the nutrition to the fetus. Okay, so very early absorption. Okay, within the cytoplasm, from the cytoplasm, then from the tubal and uterine secretion. Okay, so basically this is the histiotropic transfer from the decidua. Okay, before the uteroplacental circulation, and the hematotrophic transfer and the hematotrophic transfer, that is active and passive transfer of the fetal circulation. Okay, so basically oxygen they diffuse passively, but certain things, okay, they get active transfer. Okay. So remember, almost two-thirds of the total calcium, okay, and 3/5 of the total protein, and almost 4/5 of the total iron they drain from the mother during the last 3 months. Okay, so that's why last 3 months, okay, from 28 weeks beyond 28 weeks, it is very, very important. Okay, so that patient is not anemic, to make sure that the total iron are drained from the mother. Okay, to make sure mother is not anemic, by that's why we supplement iron supplement. Okay, similarly 2/3 of the total calcium is drained from the mother, so that is the one reason why we supplement calcium as well. Okay, and 3/5 of the total protein. Okay, So fetal blood. Okay, let's discuss briefly about the fetal blood. Fetal blood, initially hematopoiesis. Okay, the formation of the blood, it occurs in yolk sac by week 2. Okay, and by week 10, liver takes its. Okay, the hematopoiesis occurs in liver, that is extramedullary hematopoiesis, then spleen, and finally, okay, medullary hematopoiesis, it occurs later on. Okay, so by time almost majority is done by bone marrow. Okay, so initially it is slowly replaced from the yolk sac, liver, spleen, and then finally bone marrow. Okay, so erythropoiesis. So initially they look megaloblastic. The cells are immature and megaloblastic, and later on it becomes normoblastic. Okay, and the number of RBC, they are a bit more when we compare to the adult. Okay, so they are, it looks like polycythemic, but it's normal for them. Okay, so for fetus, the hemoglobin, the RBC is around 5 to 6 million, and hemoglobin is around 16.5 to 18.5. Okay, so it's bit in the higher range, upper range, because they need to extract all the nutrition. Okay, so to carry oxygen as well as nutrition. And erythroblast around 10%. Okay, so retics 5%, erythroblast 10%, because there is more division of the cells, rapid division of red blood cells. And what about the fetal hemoglobin? So initially common the main hemoglobin in the fetus is, yes, it is fetal hemoglobin, that is it contains 2 alpha and 2 gamma chain. Okay, so this is fetal hemoglobin. Okay, and after 20 weeks, so adult hemoglobin, it appears, okay, which is 2 alpha and 2 beta, but almost 80% is by fetal hemoglobin. Okay, and at term, so at term, when we measure the hemoglobin type, then it's 80% it is fetal hemoglobin. Okay, and just 10 to 20% is around adult hemoglobin. Okay, so by then, after post delivery around 6 to 12 months, it is replaced by adult hemoglobin, that is 2 alpha and 2 beta. Okay, and lifespan is also less in case of fetal hemoglobin. Okay, so that's why it is just 80 days. In adult hemoglobin, it lasts for 120 days. Okay, so these are the different. You know why? Basically, there is a abundant fetal hemoglobin because it has very good oxygen, increased oxygen affinity. Okay, so and it tries and it binds to more oxygen molecules. Okay, so that's why in fetus there is fetal hemoglobin. Okay. Okay, so let's discuss about the leukocytes and fetal defense. So leukocyte, it occurs, yes, it is seen by second month of gestation. Okay, around 8 weeks of gestation, and blood cell count, white blood cell, it raises to 15 to 20,000 per millimeter cube at term. So okay, at term, it's bit in the higher range. Okay, than adult. Thymus. Okay, initially, the lymphocyte, they are produced by thymus, then follows the spleen, follows the production of lymphocyte, and the lymphocyte, B lymphocyte, they form the antibodies. Okay, remember this is very, very important. Maternal IgG, okay, immunoglobulin Z crosses the placenta from 12 weeks onward, giving the passive immunity. Okay, so remember Z goes. Okay, remember Z means Z goes to fetus. Okay, remember IgG goes to fetus, but IgM, no, it doesn't cross it because it is a massive, very big molecule. Okay, so it do not cross the placenta. So if we detect if we detect IgM of maybe some virus, okay, or like if we suspect some infection in the fetus, okay, so let's compare one, the rubella virus. If we get IgG of rubella, then it may be a passive transfer from the mother. Okay, but if we detect IgM rubella in case of fetus, that means the fetus is infected. Okay, so it helps us in the diagnosis. So IgM, it never crosses the placenta, it's the Z that goes. Okay, okay, so another system is a fetal renal. Okay, so in fetal renal, by the end of first trimester. Okay, so by 12 weeks, the nephron start to secrete urine. Okay. So, and to near term, it's around 650 ml per day. Okay. So basically, the kidney, it regulates the composition and volume of the liquor. Okay. So main function of the kidney here is to maintain the volume of liquor, because all the other waste secretion part, okay, so that is done by placenta. So placenta is all, it takes the function of kidney as well as liver as well as lungs. Okay. So main function of fetal kidney here is to regulate the liquor volume. Okay, amniotic fluid volume. So when there is oligohydramnios, means when the liquor is low, so we may suspect there's something problem with the kidney. Okay, so it is usually associated with renal hypoplasia or obstructive uropathy. Okay, so renal hypoplasia or obstructive uropathy. That's why when there is this liquor volume, it gives indirectly an idea about the renal condition. Okay, fetal kidneys. What about integumentary system? Okay, integumentary system. So by 16 weeks, the lanugo, it's the downy hair. Okay, thin, colorless, fluffy hair. Okay, so it's usually present, and at term it disappears. So when we get lanugo in the fetus, so think of the preterm delivery. Okay, usually in the preterm fetus, you can appreciate the lanugo, this downy, fluffy hair, but in term, you cannot appreciate it. Okay, it disappears. And by 20 weeks, sebaceous gland, they form. So initially, first gland to form is sebaceous and later sweat glands. Okay. And these sebaceous gland is secrete. Okay. And these, that is vernix caseosa. Okay. Basically, these are the secretion from the sebaceous gland. They mix with the exfoliated cells, smearing the skin. Okay. This is vernix caseosa. This you can appreciate once the baby is delivered. Okay. This butter-like whitish, this butter-like substance which is covered over the fetus. So you can appreciate that in term. And when the baby is delivered, you can see the oil, whitish part, it is covering. So basically, these are the vernix caseosa. And this part is also important. Okay, so horny layer of epidermis is absent before 20th weeks. Okay, so which favors the transudation of fetal capillaries into liquor. So remember, after 20 weeks, its major contribution is from the urine, because before 20 weeks, the skin, from skin also, some part, okay, of the liquor, amniotic fluid is contributed, okay, from the transudation. Okay, by transudation, some of the fluid it is secreted, okay, from the fetal capillaries, it contributes to the formation of liquor, amniotic fluid volume. But after 20 weeks, it becomes cornified. Okay, it becomes cornified. So the horny layer of epidermis appear, and no more transudation occurs after 20 weeks. Okay. Okay, now let's discuss about the fetal gastrointestinal tract. Okay. So fetal gastrointestinal tract. So around 10 to 12 weeks, the fetal swallows amniotic fluid. Okay, so by almost 12 weeks, okay, so fetus swallows the amniotic fluid. Okay, so we have less than 1 minute in this Zoom timing. Okay, so if we get lost, then again rejoin. Okay, to continue the lecture. Okay. So fetal gastrointestinal tract physiology, let's discuss. So by 10 to 12 weeks, okay, the fetus swallows amniotic fluid. Okay, and by 20 weeks, the meconium appears. Okay, so this is the stool. Okay, so what is the composition of meconium? It's mainly the hepatic secretion. Okay, mainly it is hepatic secretion. That's why it is greenish and black in color, because of the pigment biliverdin. Okay, this is mainly the hepatic secretion. Okay, it is mainly the hepatic secretion, and other content includes lanugo here. Okay,